Today, one third of the world's population is infected with tuberculosis. And every year, 1.3 to 1.5 million people, they die from tuberculosis. So we have developed a method called TB RAP, we called it as. So this kit can diagnose TB very rapidly.
We would like to know about the drugs that you have developed for the anti-leukemia treatment, how they are helping the society and how you have introduced them.
One day I just visited one pediatric oncology ward, and there I saw small kids. They were suffering from leukemia. If you include this drug in chemotherapy, it increases the survival rate of patients to more than 80%.
We have got a kind of information about one of your activities, which very few people might be knowing about it. I think you're guessing it right. Hello everyone, welcome back to the InnoQuest Talks 1.0, where intellect meets innovation and thinking fuels invention. Together, let's celebrate the journey that begins with curiosity, grow with dedication, and create impact beyond the laboratory and classroom. Today's guest is someone whose work touches lives in the most meaningful way, through the science that heals. And so, we are honored to host Professor Avinash Sonawane, a distinguished professor in the Mehta Family School of Biosciences and Biomedical Engineering at IIT Indore, and he is currently the convener of School of Innovation also. Professor Sonawane also serves as the Humboldt Visiting Professor at the Leibniz Lung Center, Germany, reflecting his strong international academic and research engagements. His academic journey includes a PhD from Germany, which is followed by the postdoctoral research experiences across leading institutes in the USA, Germany, Norway, Japan, Sweden, and Switzerland also. He also holds multiple prestigious national and international fellowships. His research primarily focuses on two critical and impactful areas of public health and translational research. The first one is understanding pathogenicity, drug development, and the diagnosis of tuberculosis, whereas the second one focuses on the development of therapeutic drugs for leukemia. Notably, Professor Sonawane has developed and transferred two breakthrough anti-leukemia drug technologies to the industry, which we will be discussing in detail during this session. In addition to that, he has also developed novel drug candidates against multi-drug resistant tuberculosis and a food safety enzyme formulation called acrylase, which is aiming at reducing the cancer-causing compounds in processed foods. Like his innovations have been recognized with several distinguished national and international awards and honors. With over 15 years of academic leadership, which includes a leadership position of Dean International Relations at IIT Indore, he has made significant contributions in advancing research culture, entrepreneurship, collaborations, and institution development. Beyond these remarkable academic achievements, what truly stands out is his dedication to social responsibility. Professor Sonawane actively mentors students from rural and remote areas, personally visiting schools and villages to guide young learners. He's also supported many students from the economically weaker backgrounds. All of these efforts collectively reflect not only his scientific excellence but also his compassion, humility, and commitment to the societal upliftment. When we will be talking about his social responsibilities, what he carries on his shoulder, we will be discussing them in detail during this session. So let's welcome Professor Avinash Sonawane to InnoQuest Talks 1.0 and listen to his inspiring journey, his work, and all the inspirations behind his research work and his social activities. So welcome to InnoQuest Talks 1.0. We are very glad to have you here today. Thank you for taking out your time for this session. Before we dive deeper into your research work, we would like to know about your journey from Jalgaon to Germany and across the global research institutions. How was it? We would like to know that.
Thank you. Before I start to share my journey and experiences, I would like to thank you for inviting me for this InnoQuest talk series. And also, I would like to give a big congratulations to R&D team for taking this great initiative. It is helping us a lot to connect with the scientific community and the society. Before I come back to your question, I would like to give some brief background about me. I was born and brought up in a very tiny remote village. That time the population was around 200 and now it is around 500. So you can imagine how this small village it is. And I come from a farmer's background. My father is farmer. He has studied up to 9th standard. My mother has studied only up to 7th standard. My siblings have studied only up to 10th standard. So you can imagine that there is no educational background from.
Yeah, we understand that.
And I did my primary education in a village. And then after my primary education, I went to Jain College where I did my graduation. And after completion of my graduation, I went to North Maharashtra University where I did my post-graduation in microbiology.
Okay.
And this is where I got introduced to microbiology.
Okay.
After completion of my post-graduation, I went to Germany and I did my PhD from University of Marburg, which is a very old and very famous university. This university has produced almost 10 Nobel laureates in chemistry and medicine.
Oh, that's wonderful.
The department I was doing my PhD, there we had a gallery of one of the Nobel laureates, Professor Emil von Behring.
Okay.
He got a Nobel Prize in medicine in 1901 for his discovery for diphtheria antitoxin. And in that gallery, his whole journey was mentioned, that how he discovered this antitoxin and how he got fascinated about the microbiology. And I think that was the starting point for me to generate interest in microbiology.
Okay.
So that time, this microbiology was very newly introduced subject. It was a lot of fascination was there. We were very fascinated to see how these microbes they look like under the microscope.
Oh.
How these microbes they cause disease. So it was very new field for us also, and that's how we got introduced to microbiology and got fascinated about this subject. And after completion of my PhD, then I moved to USA and did my first postdoc from there, from University of Florida. There actually I started to work on the medicine. So I was working on that, how some microbe infection change the immune response of the lungs. So I was working with the patients, cystic fibrosis. They are very much susceptible for the Pseudomonas infection. And after completion of my postdoc from there, I came back to Germany and did my postdoc at Max Planck Institute for Infection Biology in Berlin.
Okay.
And there I got introduced to Professor Stephen Kaufman. He's a very famous immunologist. And he was highly cited immunologist for several years worldwide. And his group was mainly working with tuberculosis.
Okay.
And that's where I got introduced to the infection biology field. So I was having interacted with him for several times. I was following his work, and I really got fascinated that how deadly this is and what are the different challenges that are there in TB. And that's where I started to work on tuberculosis. India is also a high TB burden country. So I wanted to contribute something in developing some therapeutics or diagnostics for tuberculosis. So after completion of my postdoc, came back to India, and I started my own research group. And while doing some work, we used to visit hospitals. And one day I just visited one pediatric oncology ward, and there I saw small kids. They were suffering from leukemia. And I could see the pains that the children were suffering. And it was mainly due to one of the drugs which was given to these kids. And that's how I thought, can we work on the leukemia and develop something which will improve the treatment of leukemia. So this is how I got introduced to the microbiology and the infection biology.
Listening to this journey, it is really very interesting how you moved from a small town or small village to Germany and then across the global research institutions, and how you have got introduced to multiple things, specifically microbiology. What I could understand from this one is your curiosity didn't stop here and you continued your research furthermore. So moving towards your research work, we would like to know about the drugs that you have developed for the anti-leukemia treatment, how they are helping the society and how you have introduced them. I think that technology has already been transferred to the industries. So we would like to know about that from you.
We have developed two drugs, asparaginase drugs, which can improve the treatment of primary and relapse leukemia, particularly acute lymphatic leukemia. Acute lymphatic leukemia is a type of blood cancer, and this is the most common cancer identified in human being. So it accounts almost 70 to 80% of total cancer. And this cancer is more common in children below 15 years of age. So it represents almost 23% of all cancers identified in children. And in India, every year, almost 50,000 cases are identified of ALL. So you can imagine how the widespread and deadly disease this is. We have been working on developing this drug from last 17 years. So we started to work on this drug in 2009. And there is no substitute because this asparaginase is a very important component of leukemia therapy. If you include this drug in chemotherapy, it increases the survival rate of patients to more than 80%. And if you take out this drug from the chemotherapy, then the survival rate goes significantly down. And there is no substitute for this enzyme. But the major drawback is that when this drug is given to the patients, so the patients, they suffer from a lot of side effects, very serious side effects. It has very high neurotoxicity, so that causes toxicity to the brain. It causes toxicity to the liver. Another major drawback is it is highly unstable in blood. So patient has to take a repeated doses of this drug, and that causes toxicity to many different these vital organs. Another major drawback is these drugs, it does not work in relapsed ALL patients. The reason is this drug has, we call it as immunogenicity property, because this drug is derived from bacteria and it is given to human. So human body does not accept it. So it starts to produce antibodies against this drug. So when there is a relapse of leukemia, the patient body, they already have these pre-existing antibodies. And when this drug is given, these pre-existing antibodies, they go and bind to this drug and neutralizes its effect. The treatment of relapse ALL patients is a big challenge. So like this, there are several disadvantages, serious side effects of this drug. We wanted that how to improve the therapeutic property of this drug, and can we reduce these side effects?
Side effects, yeah.
We developed a new drug. So asparaginase is a protein, and we did almost 12,000 different changes in this single protein. Yeah. For almost 4 or 5 years, we are doing different changes in that protein and trying to see each change, does it improve its therapeutic properties and reduce these side effects. So like that, we have created almost 12,000 different variants, we call it. And then we studied each variant and looked for the desired therapeutic properties. And then finally, we found two variants which had some 3 changes in that. And these 3 changes, it could improve its therapeutic properties and reduce its side effects. So what work we have done and what we have shown so far, that the 2 drugs which we have developed, they have significantly reduced side effects. Like it is highly stable in blood, so that the patient does not need to take repeated doses, and that will reduce the toxic effect on the vital organs. Our drugs does not have any neurotoxic effect
On
on the brain.
Yeah.
It has significantly reduced elimination rate, because normally what happens when we take these drugs, they are rapidly eliminated from our body before they cause their effect. So our drug is very slowly eliminated from the body. So the patient doesn't need to take repeated doses of these drugs. And most importantly, what we have found that our drug does not bind to the pre-existing antibodies which are present in the cancer patients.
Okay.
And this will improve the treatment of relapse patients, which is the major drawback in other drugs. These are several advantages that our drug has.
Yeah.
We have developed 2 drugs like this. So one drug can be used to improve the treatment of primary leukemia, and the other drug can be used to improve the treatment of relapse leukemia.
Okay.
And importantly, what we found that, so we did a comparative study with the existing drugs, and what we found that when our drug is given to animals, it can kill more than 90% of the leukemic cells.
Oh.
Whereas other drugs, they kill only 40 to 50% of this.
So there is a huge difference.
Huge difference, yeah.
Almost twice.
Yeah. These are the several advantages that our drug, these both drugs has. And now we have transferred these both drugs to the pharma company, DK Biopharma company.
Okay.
Now we are doing this manufacturing, upscaling. Now we are setting up a facility to manufacture these drugs in GMP facility. And we have also done this preclinical trial batches. Now, very soon we are going to start these clinical trial batches and then clinical trials. So we hope that we'll be able to produce a drug which will have significantly reduced side effects and maybe at a very affordable price.
This is a great gift to the medical field, as well as, a greater contribution to the humanity. So, on the similar lines, you are also working on the food safety enzymes formulation, which is aiming at reducing the cancer-causing compounds in the processed foods. Yeah. So, we would like to know about this particular innovation of yours, especially when we are very much concerned about what we consume today. So we would like to know about that from your side.
Sure. So this is a second innovation from our lab.
Okay.
This has application in food industry. Today, this consumption of processed food has increased significantly.
Yes.
And that also puts us at the risk of developing cancer. The processed food, which are rich in carbohydrates and starch, and when these food products are processed at high temperature, so that converts these carbohydrates and starch into a carcinogen called as acrylamide. And this acrylamide, it induces a cancer development in human. We were working on developing a formulation to reduce this acrylamide production in processed food.
Okay.
So we had developed a formulation which is, it's again an enzyme-based formulation, which is active at a very high temperature. And it remains active at high temperature for longer time. When these processed foods, when we add this acrylase formulation before the heat treatment, it reduces the percentage of acrylamide to more than 90%.
Okay.
Yeah. And we have already tested different processed food products. And in all these food products, we have shown that the addition of this acrylase formulation, it reduces this acrylamide production by more than 90%. We hope that this will be very useful in reducing the risk of cancer development when you add this formulation in the processed food. We already filed a patent for this. Now we are in discussion with some food industries to take this formulation forward.
I mean, for the tech transfer that you are discussing with few of the industries. What I understand from this one is if the food industries would be using this enzyme formulations in the processed food, that would reduce the risk of cancer-causing compounds in that one. Then we may consume it freely without any hesitation. That is what I understand.
Yeah, I mean, still, we have to do validation studies, so still, some study needs to be done. But we hope that
that would happen in future. Okay, so that
hope.
will be beneficial for us. Moving to another piece of your research work, which is addressing the diagnosis and the treatment of tuberculosis. As we know that tuberculosis is still a global health challenge that we face. So your research work is addressing this, the diagnosis and the treatment of tuberculosis. So can you shed some light on this?
Like cancer, tuberculosis is a major global problem.
Yes.
Today, 1/3 of the world's population is infected with tuberculosis. And every year, 1.3 to 1.5 million people, they die from tuberculosis every year globally. India is also a very high burden country. So there are several pockets in each state. For example, in Tamil Nadu, several northeast states. Even in Madhya Pradesh, there are many pockets of tuberculosis burden. So there are two major challenges of tuberculosis. One is the treatment and a diagnostic.
Okay.
And this problem has become more serious after emergence of this multi-drug resistant strain. Now we have multi-drug resistant strains, extremely drug resistant strains, and totally drug resistant strains.
Okay.
And treatment of these multi-drug resistant strains is a big problem because many of these conventional drugs, they are not effective against these drug resistant strains. Therapeutics of tuberculosis is one challenge.
Okay.
And the other one is the diagnostic. Even today, 35 to 40% of the TB cases, they remain undiagnosed.
Oh my God, that's a huge number.
That's a huge number, yeah. And these undiagnosed people, they become source of transmission of this disease because it's an infectious disease.
Yeah.
And if the person is not diagnosed, so that becomes source of transmission of this disease from one person to another person. So development of early diagnostic methods is very, very important. Our group is working on developing some novel strategies to improve the diagnostic and therapeutics of tuberculosis. In terms of diagnostics, so there are already several methods are available for TB diagnostic. But these methods, they have several limitations. Like some of these techniques, they are very time consuming. It takes 6 to 8 weeks for diagnosis.
Okay.
Some methods, they are very expensive. Their sensitivity is less, specificity is less. And many of these techniques, they require a sophisticated labs, equipments, and also the skilled manpower to perform these tests. And tuberculosis is a poor man's disease. It's not mostly predominantly found in rural
and
areas.
remote areas. Yeah. That is what you find.
So we need technologies which can be easily deployed in these low resource settings areas or in remote places. Keeping that in mind, we have been working on developing a diagnostic kits which can be easily deployed in rural areas. So we have developed a method called TB RAP, we called it as. RAP means rapid.
Okay.
Yeah. So this kit can diagnose TB very, very rapidly. So this TB RAP kit can diagnose TB from non-invasive samples, for example, from the sputum, from saliva, from tongue swabs. Okay. So patient just needs to give a saliva or sputum or tongue swab, and we can detect TB from these non-invasive samples.
That's good. Just like the one kits that we were using for COVID-19.
COVID-19 or also a pregnancy kit, for example.
Okay.
This TB RAP kit is what we have done study in a small TB positive population. And what we found that this TB RAP kit, it can detect TB within 3 hours.
Oh.
Yeah. It does not require any sophisticated instruments or lab.
Okay.
Is sensitivity is more than 98%. Is ultra specific. It does not require any skilled manpower. Okay. Because that's a huge challenge in rural
remote
areas.
Yeah.
In this kit, you can just take a saliva or even tongue swabs, and then it takes processing takes 10 to 15 minutes, and then you put these processed samples in a reaction mixture that takes 30 to 40 minutes.
Okay.
And then that reaction mixture, you just put it on the strips, and that will give a color just like a pregnancy kit. So in 3 hours, you can detect a tuberculosis.
That's a huge difference. 3 hours are, and then 8 to 9 months diagnosis. So that there is a huge difference here.
But the most important thing is that this kit does not require any sophisticated equipments. We just need one heating block.
Okay.
Yeah, and it also does not require any skilled manpower to perform this test.
That is the USP of this.
That is the USP of this. Yeah, I think this will be a good addition to have such diagnostic platforms in remote areas.
Yeah.
So this is one kit which we have developed. On therapeutic perspective, as I mentioned earlier, the treatment of this multi-drug resistant strains is very, very challenging, because most of these first line and second line anti-TB drugs, they are not effective against this extremely drug resistant and totally drug resistant strain. Okay. And it's mainly because most of these drugs, they have a single target. We are trying to develop anti-TB molecules which can have multiple targets.
Okay.
So we are collaborating with Professor Venkatesh Chelvan from the chemistry group. Okay. Chemistry department, and also a few other groups from other institutes. So we're trying to develop some hybrid molecules which will target different proteins in mycobacteria, so that we can improve the treatment of this multi-drug resistant strain. So in collaboration with Dr. Venkatesh, we have already developed one hybrid molecule which inhibits the synthesis of one polycatride synthetase enzyme, which is very important for synthesis of the cell wall of this bacteria.
Okay.
And if you inhibit this enzyme, there is no synthesis of the cell wall, and as a result, the bacteria, they die. So we already got one US, China, and Indian patent for this, and now we're doing some preclinical studies on this drug.
Okay. Yeah. We would like to go to your lab. If you allow us, then we would like to see some visuals from your lab related to all of these technologies that you have developed and as discussed so far. So would you like to take us for a tour to your lab?
Sure. Yeah. So we'll go and see that how the students are working. Now, of course, whatever work I have told so far, it is not just me. It is a team effort.
Yes.
My
your
PhD
research
students,
group is working.
my collaborators.
Okay.
So the whole credit goes to my research team and my collaborators for contributing in developing these molecules.
Oh, yeah. Thank you so much, sir. So let's go to your lab. We will have the visuals of it. Thank you, sir, for taking us to your lab and showing the visuals of your research work. Now, coming back to our discussion, what I could understand from the past discussion is that there are two technologies which are available with you, which are supposed to be transferred to the industries. One related to the tuberculosis diagnosis and treatment, and the second one is food safety related technology. So both are supposed to be transferred to the industry. That is what I believe. Yeah. So there is a chance for the industries to collaborate for this pieces of your research work. That is what we believe. So the healthcare industries can reach out to you, and they can take the advantage of collaboration for this pieces of the research work. What today's world need is translational research, which is the research from lab to market. And it is not focusing only on the research being carried out in the lab or publishing the papers or just filing the patent, but all the research work is supposed to be taken out from lab to the market. And you already have done for two of the technologies. So we would like to know what are the challenges that you have faced during this tech transfers.
I think you need to arrange another episode to
listen to this
the challenges what I have faced in developing this technology.
Definitely, sir.
Or I can write one book that the challenges which I have faced while developing this drug and for this technology transfer. As I mentioned, we started to work on this drug for almost 17 years back, and I also learned because this drug development and technology transfer was very challenging.
Yes, of course.
But it was very fascinating experience for me also. I also learned a lot because doing research in the lab is one thing, and developing some product and taking it to the market is a different thing, and that's really challenging. And probably this is the reason why not so many drugs are developed in our country, because it really takes a lot of time. When we were working on developing this drug, we faced many challenges right from the beginning, because drug development is a very expensive affair. So it requires a huge funding. And there are multiple steps are involved in developing this drug. You cannot get just one project which will fund all the work which is required for drug development. So at each step, you need a funding source. And I'm quite fortunate that I could maintain that continuous funding for last 15 years when we were developing this drug. So there are several challenges while developing some technologies, in particularly the drug development. One is the funding, because drug development, it requires a huge funding. So you need to have a continuous source of funding to take this product from one step to another step. The second challenge is to have a sophisticated facilities, because we are developing a drug. It has to be manufactured in a very controlled environment. So you need a GMP facilities, that is good manufacturing facilities to manufacture this drug. You need facilities for upscaling to the industrial scale and getting highly pure product. Setting up that facility is itself a very expensive stuff. You need patience, of course, because as I said, it takes decades sometimes to develop these drugs. So you need a patience. That is, I think, the most important. Another major challenge is to identify the right industrial partner. I'm not saying just identifying the industrial partner, but identifying the right industrial partner is very important. It's mainly because the researchers and industries, they have very different thought process. We as a researcher, when we develop something, we think that, I have developed drug or I have developed this technology. But when you will start to discuss with the industries, because where the researcher ends, thought process ends, that's where the industries, they start. Developing is one thing, but taking it to the next level and then marketing is a very different this aspect. You need to identify a right industrial partner. I have when we developed this lead molecule, this leukemia drug, I discussed with at least 25 pharma biotech companies
Oh my God, that's
for
huge.
different types. And it has happened that sometimes we discuss with the industries for almost a year or maybe more than that. And then at the end, they told us that we are not interested. So that takes, like convincing, identifying the partner and convincing the industries is a major bottleneck, I would say. But fortunately, we found one pharma company, like who has shown interest in our product, and now they are taking this product to next level. When we are discussing with the industries, and when we start this negotiation with them, the industries will normally try to show that like your product is good for nothing.
Oh my God.
Yeah. So, but you need to have confidence in your product. Okay, so whatever I have developed, it has a potential.
Yes.
Never get disappointed when industry says, because they have their own interest. Never get disappointed when industry says that your product is good for nothing. You should believe in whatever work you have done. Establishing the facilities to produce this product at the large scale is another challenge. But while negotiating with the industries, I think the most important thing is to do a proper value this audit of your product valuation. We call it, because sometimes your product may have huge potential, which you are not aware. Not only in India, maybe in some other countries. Knowing this potential of your product is very, very important.
Yeah.
And before you start discussing with the industries, don't forget to protect your rights, your IPR. I think that is very important.
Very much.
So normally, like whenever we go and discuss, we tell so many things openly.
Yes.
But I think when you like are discussing with the industries, I think this is very, very important before you start negotiations with them. Protecting your rights is very, very important.
Yes. Yeah. Thank you for sharing the challenges that you have faced during your technology transfer. This information will be very much useful for the researchers and scientists who are looking at the translational research. I mean, taking their research from lab to the market. That will be very useful for them. Now, moving to the next very important chapter of your journey, your leadership role as Dean International Relations. We would like to know about your experiences. How was your experience as Dean International Relations at IIT Indore? And we would also like to know about your strategy behind the international collaborations that which are done so far.
I must say that I had a very enriching and a memorable time as Dean International Relations. And thanks to Professor Nilesh Jain, who has appointed me as a Dean International Relations first. And then thanks to our director, Professor Joshi, for his constant support and guidance. And I think because of his constant support and guidance, we could put our institute on a global scale. Okay. And today I'm very happy that I could contribute a little bit to increase the visibility and the global networking of our institute. So I took a charge of Dean in 2020, just 2 months before the COVID.
Oh my.
So after 2 months, the COVID started. And for almost 1.5 years, we couldn't do much. Already some foundation was laid for the international office. But my objective was to increase or to take our internationalization to the next level and to increase the visibility and the network of our institute at global level. So we took several initiatives to achieve our objectives. Our first goal was to involve the participation of the faculties and the students. Okay. So we started several programs where we made faculties as the in charge of these programs. In some cases, we made them coordinators of some programs. So that has helped us to connect with the faculties. And today I'm very happy that a large number of faculties now they are associated with the international office. And they are contributing in various capacities. Same students. So we form the Students International Cell. So it's a representation of the students, and these students actually they were acting as a bridge between our international office and the students, and they organized several activities that has helped to increase the participation of the students in various activities. Okay. And then while doing this, we had discussions with many universities across the globe. So we organized various meetings, workshops, regular interaction with the universities. And today, I'm very happy that we have networked with more than 300 universities across the globe.
Oh.
Yeah. And during this time, we took several initiatives. And it's again my big thank to Professor Joshi, our director. He has supported all these schemes. So whenever I went and talked to him and proposed some new initiatives, he readily helped and supported these activities. So whatever we could achieve today, it's mainly because of his support. We took several initiatives, we visited several universities. So we used to take a delegation from our institute and then visit several universities, top-ranked universities in that country. So we used to visit different departments, talk to their faculties, see how we can collaborate with them. Then we started different fellowship programs for the students. So there are almost 50 fellowships are given by the institutes to the students, both for inbound and outbound students. So for PhD students, we started this 3 months PhD fellowship program, where our PhD students, they can go and spend up to 3 months in any foreign labs. Similarly, any foreign PhD students, they can come and spend up to 3 months in our institute. We have another program for undergraduate and postgraduate students, a semester exchange program, where the students from both sides, they can come and spend up to 1 semester here. So this has really helped to build our network with the foreign universities. Similarly, we also started some programs for the faculties, like international travel grant, because there are many new faculties that are joining here. So they need some support to develop their collaborations with the foreign universities. So through this international travel grant, these faculties can go and spend up to 1 week in any foreign lab. And they can discuss with their collaborators and come up with some joint projects. Similarly, we have started this mobility grant for the faculties, because we are signing a lot of MOUs with foreign universities. But actually, to make these MOUs more effective, we made several faculties as the MOU coordinators. And we requested them to organize different activities under that MOU, like organizing conferences, mobility of the students, workshops. And I think that has really helped to increase our activities. Okay. And I think because of this, several initiatives, today we have signed more than 130 MOUs with more than 30 countries.
Wonderful.
The universities. The number of foreign students has increased significantly. Similarly, the number of applications, we are receiving good number of applications from many different countries. It's mainly because of our whatever networking which we have developed with these industries. So I think overall our global visibility has increased significantly. So I'm again happy that I could contribute in putting our institute at the global scale.
Global scale, yeah. Now, going beyond the institution building, slightly towards your commitment to the society, we would like to discuss that. We have got a kind of information about one of your activities, which very few people might be knowing about it. I think you're guessing it right. So let me tell our viewers that Professor Sonawane has supported many students from economically weaker backgrounds. He has helped them with the books, mentorships, and financial assistance also. He keeps on visiting schools and the villages to help this young mind. So coming to you, sir, what is the inspiration behind this? How do you take out your time from this busy schedule for this activity?
This is something I don't discuss much, but what I feel that we owe to the society, and I think we should give back something to the society. As I told you in the beginning, that I come from a very rural background, and I have gone through that struggle and hardship, and I know how challenging it is to achieve your objectives. So in villages, there are many talented students. They want to excel in their career, but sometimes they don't get proper mentorship or guidance. Many times they are not even aware about that. What are the opportunities to interact with the students? So some students, if they need some mentorship, I help them. If they need some career guidance, I give them this career guidance. Sometimes if they need some assistance, like some books, so I donate books to them. Or sometimes if they need some financial assistance, I help them. And I think that gives me a lot of satisfaction. And I'm very happy to tell that there are some students whom I helped. Today they are working at a very good colleges, or they got some good jobs. Okay. And I hope to continue this. So
in future also.
Yes.
So this is good to know about this activity of yours. I mean, giving back to society, that is what you believe in. Before we conclude, I would like to know if you can share a short message for the students or the researchers and scientists who are working in your research domain.
I would like to say that be passionate about whatever you are doing. Never get satisfied with whatever you have achieved in your career. There are many things to do in a life. But while doing this, be a good human being. Help to the needy students. God has given you so much. I think it is also our responsibility to give back something to the society.
Thank you for giving us the time for this session. It was really interesting to discuss your technologies, your journey, your social activities on this platform. Thank you so much for your valuable time, sir. Thank you for being.
Thank you very much. And before we conclude, I would like to. I really liked your tagline from Jalgaon to Germany.
Germany. Yes. Oh, thank you. Thank you so much, sir. Professor Sonawane's work stands as a powerful example of how science guided by the purpose and empathy can truly change the lives. And to our viewers, thank you so much for tuning into this episode of InnoQuest Talks 1.0, where Professor Sonawane discussed his research work and his journeys so far. If you are interested in research collaboration opportunities, please feel free to reach out to us via the email displayed on the screen. InnoQuest Talks 1.0 will continue to bring more engaging discussions on research, innovations and technologies at IIT Indore through our YouTube channel, Indian Institute of Technology Indore Official. On this channel, you can check out our episodes related to the InnoQuest Talks 1.0. Until next time, keep innovating and keep exploring. Thank you so much.